Okay.
Everybody, this is Amanda Sierra on behalf of all the organizers from this series, Rosa and Claire, Gloria, Giorgio, Fede, and Marta. Today we have an amazing speaker. We have Elvira Mass from the University of Bonn. I just want to remind you of the following talks that we are going to have with really great speakers too coming up here. So please join us. Also remember that you can join the list, the email list. Just send an email to any one of us or the link that you can find here on the website, and we will put you in the web list and the email list, so you will receive all the emails. So I'm just gonna leave the room to Fede Soria, who will be introducing Elvira, and then the questions will be handled by Gloria Colombo. So welcome, Elvira. Thank you very much for joining us today.
Okay, hello everyone. Today we have Professor Elvira Mass. She's a professor at the Life and Medical Science Institute in the University of Bonn. She got her degree in 2009 in biology in the University of Bonn, and then her doctorate in 2013 in molecular biomedicine. Then she did a postdoc at the lab of Professor Frederick Geisman, first in the King's College in London, and then at the Sloan Kettering Cancer Center in New York. She has received several awards, like the ERC Starting Grant and several Young Investigator Awards. Her research focuses on understanding the diverse roles of macrophages and in tissue homeostasis and disease. Her research underscored the critical roles of macrophages in establishing the diverse physiological and pathological immune states during tissue development, the immune response to metabolism, and during neurodegeneration. Thank you very much, Elvira, for this talk, and the floor for the screen is yours.
Yes, thanks a lot to the organizers for inviting me here also to kick off the season, let's say, with a talk on microglia and one of my favorite cells, and also today on T cells, a new cell that I'm trying to understand, and I hope that you will bear with me today, since it's the very first time I present the whole story, and I'm very happy to take any input and ideas from your side as well. So today I will talk, as I said, about microglia and T cells and neurodegeneration, but as I was introduced by Fede, we are actually very much interested in macrophages in general. And here we are interested in the origin of macrophages, and most of you may know that microglia have been among the first macrophages described to have a York-like origin. So they stem from erythromalid progenitors. And with the precursor state of a pre-macrophage, which we described, they will give rise to microglia very early during embryogenesis. However, the brain can also, of course, carry other types of macrophages, here called CNS-associated macrophages or border-associated macrophages. They can carry different names. And the current state of knowledge is that there may be some also long-lived and so really long-lived macrophages outside of the parenchyma, but also some populations that are mixed, and also monocyte-derived macrophages, so coming from the bone marrow all the time and being replaced. And this is actually not only the case in the brain, but also in all the other tissues in our body, that we have these different types of origins, but also the longevity, which is less well understood in outside the brain. What is also interesting is that we have in the in the brain, we also have other immune cells that can be long-lived, and these are tissue-resident memory T cells. And this is less well understood than microglia, and I will just briefly give you an introduction on these cells. So we have here tissue-resident memory cells that can be defined by markers and their antigen experience. Depending on the residence markers they upregulate, you can define them as resident, but they can also express certain receptors that will basically crosstalk to myeloid cells, to microglia and macrophages, as well as chemokine receptors to respond to the, let's say, neurodegeneration, neuroinflammation signals that come from the brain. We also have central memory and effector memory T cells that are circulating in lymphoid and non-lymphoid tissues across our body. And people have performed parabiosis experiments, as well as injection of antibodies to really define these different subsets and to look at these cells. And what we know today is that we have in young and let's say clean SPF mice, we have very few T cells in the central nervous system. However, with aging or with any kind of disease, infections, we will have experienced CNS with an increase of T cells at different sites of the brain, not only the meninges, but also in the parenchyma. There are cells found in different kind of diseases. And of course, the microglia field has really profited from different fate mapping models in the past to really study the impact of microglia in neurodegeneration. And here, the lab of Stefan Jung has really made seminal published seminal works here on fate mapping, but then has been used in different kind of diseases. And here, for instance, we have the CX3CA1-CRE that really is helpful to map microglia and then look whether monocyte-derived macrophages will enter the brain later on. And now we also have new models that can not label all macrophages microglia using this CRE, but also very specific using the split CRE approach. So we have two CREs basically working together to label microglia or other border-associated macrophages such as perivascular macrophages. And this has been very well studied, or is now a lot studied in mice, especially and in its context of ontogeny and what this microglia are doing. But what we were interested in is what are actually incoming cells doing. So we have the response of microglia, that's interesting, but we also need to know what is coming in, and even though it may be transient. And this question was asked by Ralf Stumm. He's working at the University of Jena, and he's actually a neuroscientist. He works on interneuron migration during development. And this is why he also worked on this molecule, CXCR4-CREATE2. And then he came to us a long time ago and said, look, I think this model is really good to look at infiltrating cells into the brain, because I do not see any microglia labeling if I inject tamoxifen 5 times in a row. But I do see a lot of cells coming into brain being labeled. So we studied with him within the context of stroke. We studied with him the role of monocyte-derived macrophages and microglia, and this was published quite a while ago. And I think this model is really suitable to combine it with any kind of knockouts, transgenic mouse model brain injury models to study the impact of monocyte-derived macrophages versus microglia quite well. But what was also interesting is that actually the blood T cells were also very well labeled. And I will come to that in a second, which allows you really to have a broad picture of the immune infiltrates in the brain. And we chose this particular model here, which was also driven by Ralf, spastic paraplegia, or more specifically SPG15. This is just another gene name, or encoding the protein spasticine. And this was due to the tight collaboration with one of the hospitals in Jena, because they have been seeing these very rare patients and trying to understand the disease. And Ralf, being curious about the brain immune cells, he just had a look at the brain and said, microglia look very weird. Don't you want to work with me on this new project? And I said, back then, setting up a new lab, of course, let's do it. And not knowing how difficult it will become at some point. But I will tell you today what we found so far. So first of all, because maybe not all of you have heard about these diseases, they're really a very, or among the largest groups of inherited neurological disorders. And you really see behavioral phenotypes in the patients with prominent spasticity, lower extremity spasticity due to degeneration of the large motor neurons. And they're really diverse, these disorders, with over 80 distinct loci in the so-called SPG genes, spastic paraplegia genes. And just to give you a flavor of the prevalence of this disease and why it is quite difficult, maybe also to study it taking human material, is this distribution from different studies. So this is hereditary spastic paraplegias being autosomal dominant or autosomal recessive, and sometimes you just take them all, but you see roughly 3 to 5 cases per 100,000 only in different regions of the world. And if we now zoom in a little bit into the most common forms, here's the autosomal dominant form, most common. So here you have a few thousand patients, maybe in whole Europe, and SPG15, which we will focus on today, is even less prevalent. So it's quite hard to get by primary material. So therefore, people have looked at, especially in vitro cultures and ex vivo cells, to try to understand what these genes and what these proteins are doing. And here the map is quite diverse, let's say. Here are all the different genes that have been defined to cause disease. If they're not functional, and they can be involved in many different, let's say, processes in the cell. And this is not, of course, focusing on motor neurons now. So we have some genes involved in metabolism, mitochondria, but also especially the transport microtubules. And today, as I told you, we talk about spasticin, so SPG15, which is an important protein in the lysosomal autophagosomal pathway. And this forms a complex together with SPG11. So these two work together, and therefore their phenotypes are also quite comparable, also when it comes to mouse knockouts. So this is really this coordination of this complex and this complex in autophagosomes is really critical for the clearance of defective organelles, of protein aggregates, and therefore it is really important for the integrity of the axonal of the axons in the motor neurons, so that if this is not functional, you will have degeneration of the neurons. And so this has been described, but recently, and this is very interesting also then to us, very recently a paper came out looking at 3 SPG11 patients. And there you can also see, and this is an autosomal recessive form, which is most common with 20% across all recessive form, that in this postmortem brain, you can see a severe neuronal loss in the frontal cortex, also in some other parts of the cortex, especially in layers 4 and 5. So these layers basically degenerate, and you can see typically a neurodegeneration by reduced neurofilament signal, but also less myelin in these brains, and reactive gliosis is stained with the GFAP here. And SPG11 knockout mice have been published a while ago, as well as SPG15 mice, which showed a very similar phenotype actually. So here you can see again the cortex of these mice now at 16 months of age, SPG15 mice that have also a decrease of new and positive cell here in the cortex. And also here in this electron microscopy picture, you see degeneration of the large motor neurons that you can see here, basically really showing and recapitulating what we see in the patients when they have died already. And if you now dig into the especially supplemental figures of this paper, you will see that people have also looked a little bit into the immune compartment of this SPG15 knockout mice. And you can beautifully see here the whole brain, GFAP signals really increased across different brain regions, really hinting towards a gliosis. And of course, microglia are likely to play a role in that. And this was the picture commented with one sentence that there may be immune activation in these animals involved in this disease. And this is where we basically started. Our big question was, are microglia or any other immune cell contributing to the disease of spastic paraplegia, especially in context of SPG15? So first of all, what we did is we took these animals and we confirmed that these animals really have a behavioral deficit, and this really starts quite early. So already at 8 months of age, confirming the paper that I just shown you, and it really gets worse with age. And what we also could see is that the large motor neurons that are Chet positive in the spinal cords, that they're really decreased in the knockout animals, and this is quantified here. So really confirming that these animals display a similar phenotype as we would see in the patients. And I showed you before that at 16 months of age, you already see in the cortex neurodegeneration, but here we went a little bit earlier because we wanted to really understand how can, let's say, lysosomal dysfunction of a motor neuron in the axon lead to cortical phenotypes. And when we look now at early or earlier time points before the 16 months, actually what we do see is that there's no neurodegeneration in the brain yet. So in the cortex here, We looked specifically at the motor cortex at layers 5 and 6, as in the paper I've shown you before, but there is no change. And also in the spinal cord, neuron-positive neurons seem to be okay. So the big question was, now we confirmed the loss of motor neurons, but where do the changes in the brain come from later in life? So we digged a little bit deeper, of course, focusing in the beginning on microglia. And here we performed, first of all, an IBA1 staining, always in spinal cord and cortex. You can beautifully see that IBA1 positive cell, they increase even in intensity, but also in numbers. So you have really an immune activation, or if you want to call it neuroinflammation in the brain. And one typical thing that microglia do if they're being challenged or activated, they downregulate homeostatic proteins and genes, and P2RY12 is one of them. So again, in the spinal cord and cortex, you have nice expression of this protein, and this is going down significantly in these animals. And I remind you again, neurodegeneration in the brain is not yet present. So we have really an immune cell or microglial activation already ongoing before even the onset of neurodegeneration. One other hallmark of microglia typically is the morphology, showing an activated state. And also here, 3D rendering really again confirmed that something's going on in this microglia, and that loss of SPG15 function is also changing the ramification, so making the cells more round, more amoeboid. So now, having this a little bit better characterized that really something is going on in the brain of these knockout animals, we went on and looked at expression of different kind of markers that are being associated to neurodegeneration and other diseases, and one of them was MHC2. And here you can see that, and this is now the cortex, and you can see the meninges, that you will have always a little bit of MHC2 expression in the meninges, and this increases here in the knockout animals. But what is really was really interesting to see is that you have this MHC2 expression just everywhere in the parenchyma and also in the spinal cord. And this was really unexpected to see this kind of expression patterns. So of course, coming from the ontogeny part and thinking, okay, maybe the blood-brain barrier is compromised, maybe monocytes are coming in. We went back to our mouse model here and crossed it to SPG15 knockout mice. And here you can see mice that are 12 to 14 months of age that have been injected with tamoxifen at 1 month of age. So, you inject, you label, then you let them sit. And you see beautifully almost 100% of monocytes being labeled and roughly 80% of T cells in the blood being labeled. And if you now look at the MHC2 positive cells in these animals, you can appreciate here that there is no TD tomato signal in these. So this really means TD tomato indicates a monocytic origin. So everything coming from the bone marrow will be TD tomato. And this is not the case here. You see some round cells here and there, but P2RY12 microglia that are MHC2 are not TD tomato. So there's no recruitment of monocytes, and we confirmed that using flow cytometry with a bigger panel. So here we can, by the surface expression markers that are listed here, define microglia, border-associated macrophages, neutrophils, and T cells. And here in this microglia cluster, and is also quantified here, you do not see really TD tomato expression. So there's no influx. Even in the control animals with aging, you do not see a large influx of monocytes. As expected, neutrophils will be TD tomato because they come from the bone marrow, and also a part of T cells is TD tomato. And with young animals here, and we'll compare them sometimes with the old ones, these are 2 to 3 months old animals now. So 2 to 3, and these are 12 to 14 months old animals. We digged in a little bit deeper into what is actually happening in the other part that we see here in this UMAP. And if you plot the ratios of these cells, then you see that T cells are expanding. So everything else kind of stays the same, but there is a little bit of expansion here in the young animals and especially in the old ones. And this is when actually Mark Bayer here in Bonn from the DZNE came into play whom I contacted and said, look, I think we have not only microglia there could be also T cell fraction in the brain, and he's really the expert on T cells and also on neurodegeneration. Therefore, we started together with Ralf and Mark to really dig deep into this model. So when we went into the microglia a little bit more in detail, we tried to understand whether we have something that people see in Alzheimer's disease and other kind of different neurodegeneration. And we could define by marker expression again, flow cytometry marker expression, that we have what people call disease-associated microglia, because CD11C is being now expressed, MHC2 is expressed. So this are, let's say, bona fide DUMS. And we have something that we call homeostatic microglia, because they don't have MHC2 expression, and they have different kind of expression markers that we defined then as homeostatic, as a normal microglia that you will see also in young animals, especially. And what we saw is another subtype of microglia, what we called intermediate. Why intermediate? Because these cells had already MHC2, confirming, you know, what we saw in immunofluorescence as well, but they did not have 11C expression yet. So it seemed to be kind of like a, you know, transient state maybe, and that's why we call it intermediate. And when looking now at the brains of SVG15 mice, and so now young mice, we did not see a huge difference. And we also made an effort to look at different parts of the brain, because, as I said, of course, in the spinal cord, you would expect a phenotype, and maybe also at some point in the cortex. But what about the other brain regions? Where does the neurodegeneration start, and where do maybe additional phenotypes come from? And you can see a huge fraction of cells in the young animals are both in wild type or control animals and knockout animals quite high, and there are less DUMS in here. But with age, you will see an increase of DUMS in comparison. So here, the control animals in different areas, not even in the cortex, which was quite interesting, although we see, you know, IBA1 being increased in general, but it increases not in a certain population here. Instead, you have cerebellum affected, but also the encephalon, and of course, the spinal cord as expected. Trying to understand what are these cells are actually making that could drive neurodegeneration in the brain itself, we went to a single cell sequencing experiment, and we combined this with site-seq. So this is, you have barcoded proteins that you can incubate with your cells and sequence them, so you can quantitatively really define also protein levels at the same time as transcript levels. And this is now the transcriptomic data, where you can see again some neutrophils, some border-associated macrophages, and we found back our 3 populations of microglia here. And you can appreciate maybe here that the red disease-associated microglia cluster seems a little bit bigger than in the controls, as expected, as we saw also in the flow cytometry quantification. And just here to show you, again, we have the homeostatic microglia, which we defined according to normal gene expression. You would see in this kind of microglia like TMEM119, P2RY12. What you do then see in the DUMS, that you have ApoE expression, LPL, FBP1. Again, in other studies of neurodegeneration, you will find these genes again and again. So really, let's say, a bona fide DUMS signature. But then when we started to compare these clusters, so they're homeostatic to each other and the DUMS to each other, what we observed then is that actually all of the 3 populations that we had here, when we compare the knockout over the control, so it's a log-fold change. So the redder, the higher it is expressed, you can see that this typical DUMS signature genes are actually even higher expressed in the knockouts. But this is also already true for the homeostatic microglia. So it seems something like ApoE, although defined in general transcriptionally as homeostatic, it already starts to have kind of this DUMS signature. And therefore we went on and we created in silico a DUMS signature, taking cell papers into account. And here you can compare control and knockout. So the DUMS, they have the DUMS signature, they're higher in the scoring, and they're the same. However, already at this homeostatic state or intermediate state, there's a slight but significant, very significant shift here already of the microglia towards a DAM signature, meaning that even though these cells seem in general transcriptionally fine, they're already kind of shifting towards a DAM phenotype. And this is not only true on the transcript level, but also on the protein level. As I told you, we also performed site-seq in parallel, and here just 3 examples and expression of proteins. So all 3 subtypes express less P2RY12 quantitatively, less CX3CR1, but more CD11C already, which you can in this case detect beautifully in a quantitative manner. So what we concluded from this is that SPG50 knockout microglia lose their homeostatic phenotype even before the onset of neurodegeneration. And so even the homeostatic microglia, we don't know how early this starts, but at least in this 1-year-old animals roughly, they're already changing. So I told you before that we also see a recruitment of T cells. And of course, this kind of was the obvious question to ask: do these microglia kind of call T cells? And maybe this is something that could drive neurodegeneration further. And performing Go enrichment analysis of wild type versus knockout animals, we could see expected somewhat antigen processing and presentation being upregulated in the knockout animals, among also P38 MAP kinase cascade, which induces or indicates also cellular stress. And we could confirm, and this is now quite interesting, and maybe some of you have some ideas on this one, because this is now the white matter of the spinal cord. You see this accumulation of IBA1 positive microglia. There is no MHC2 signal in these particular cells, but you can beautifully see that the cells really kind of hug T cells and really interact tightly with T cells in here. And remember, this is the time point where we do see loss of motor neurons already, but they're in the gray matter. And we don't see neurodegeneration in the brain. So this is also happening in the brain, but we can see this also in the spinal cord in the white matter. And just to quantify this, this is now the old animals. You can really see an significant increase of T cells in old animals, but also if you look carefully at the young animals of 2 to 3 months, you can already see that something's happening there. So there's already a little increase there. And coming back also to the fate mapper, we wanted, of course, to see where are these cells coming from. Are they being recruited from the bone marrow? And actually, this was quite interesting. And we stumbled upon this, not knowing what to do with it, because you can see that in the knockout mice, there is not more TD tomato signal. So it's not like you have a huge influx of bone marrow-derived T cells in the central nervous system, but rather it's like a 50-50 distribution. And you have the CD3 signal here, for instance, 2 T cells that are red, but you also have one here that remains not red, which means that it must have entered the brain before 4 weeks of age. This is at least the assumption, or the, let's say, proxy we have. TD tomato means that you entered the brain before we injected tamoxifen, so in the first 4 weeks, and TD tomato is then afterwards. And if we now look at the CD8 and CD4 positive T cells, they both expand, but especially the CD8s are really expanding quite extensively. And of course, in the T cell field, like in the microglia field, there are even more subtypes of T cells. I introduced 3 subtypes to you in the beginning, but the picture is much more complex. I will just briefly guide you through that. So here we used again flow cytometry, and we went into looking at different types of CD4 T cells as well as CD8 T cells. And what you can appreciate here, and this is, I showed this to you, tissue resident memory cells here in red are increasing in old mice, but even further increasing in knockout mice. But what is even more increasing are, for instance, this PD1 positive CD8 T cells activated, or tissue effector, more T cells with an effector function. And the same is true also for the CD4 compartment. Here you can just see the split between the different ages and genotypes. And if we now look at the different parts of the brain again, we can see that in the young, the T cells are accumulating in particular regions already, maybe in the brain, but it's not that clear. However, in old brains, you can see an increase of T cells, especially in the cortex, in the diencephalon, but also brainstem and spinal cord, although not significant, not so much in the cerebellum. So just to remind you again about the numbers. So using this labeling, we can label 80% of T cells in the blood. So assuming, if we come from a macrophage perspective, if I label something around 80%, then if I have 80% in my tissue, you would assume that they all come from the blood. And I told you before that in general, all T cells are only 50 or 40% labeled. And this is now separated into different subpopulations. So here you can again see that maybe this population, which is KLRG1, PD1 positive, may be mostly coming from blood, but not only, because we're also here around 80%, but everything else, it seemed to be really that you have this accumulation that is driven also by local expansion of T cells and the recruitment of T cells. And it's really conceivable that these T cells could also drive the neurodegeneration in SPG15 knockout mice. And to try to understand that, we performed a single cell sequencing experiment on CD3 positive T cells. And you can see here the different subpopulations. We identified mainly CD8s. CD4 were too few to further subcluster them. And we mostly could really find back our subclusters or population that we saw via flow cytometry. So we have again the tissue resident cells, the effector cells, and what I would like to draw your attention to is this particular population, the KLG1, PD1 positive one, because this has really genes expressed in the type 1 interferon signaling pathway, really quite high. So really you have these cells now coming in. There are not many in the brain then, but they're coming in, really being one of the possible, let's say, communicators maybe with microglia, and that they could maybe support the microglia activation that we see in these mice. Now, trying to understand whether it's really expansion of T cells or just random kind of recruitment of cells, we took this data because it's SmartSeq data. You can also do T cell receptor analysis on these cells, and we tried to map these clones. And here are just the T cell clone ID. So here, for instance, 10 cells in the knockout have the same T cell receptor. And this really speaks for a clonal expansion of these cells. Here you have also one clone that is very dominant in the controls, but you have mostly really 4, 3, and 2 clones with 2 cells that are found in the knockout. And if you look at the distribution, which cell types you can find them, they're kind of distributed all over the place. So it's not like a particular subtype that they're made. So it's not like a cell is coming in and then clonally expanding, but rather that you have kind of this plasticity of these T cells, and they can become different type of effector cells, especially in a knockout. And when comparing now for especially these 2 clones with each other to see whether the knockout clone 13 versus the control clone would have a different transcriptional makeup, you can see that these T cells are more active actually, or this particular clone is more active than the control clone, really hinting towards the fact that the T cells in knockouts are highly activated. And I showed you before that we have many clones actually coming into the brain, and here just mapped cells that have 2 or clones that have 2 cells or more than or equals to 3 cells. So there are many here. And as I told you before, what we have is really distribution of different T cell types. But what you can see here is that in the blue, the wild type has mostly expansion of tissue resident memory cells. So the cells that are in there and resident, while in the knockout, we have more influx or local expansion of here in gray, for instance, or more of and in blue, more of the effector type T cells. Now, to understand really whether we have incoming or locally expanding cells, we took advantage of the TD tomato signal that we also sequenced in these cells. And this is mapped here. And what you can see now in the CD8 T cells is that typically, if you have a clone, it's typically only of one color. So either TD tomato negative or positive. So meaning that you have a cell coming into the brain and it's expanding or proliferating once, and only a few fraction of cells that have both, so TD tomato negative and positive. However, in the knockout animals where we don't have yet neurodegeneration, at least not visibly. You have a large fraction of clones that have both fluorescences, so being T-utomated or being not T-utomated, really hinting towards kind of like an activated T cell state in the periphery, and then they can enter the brain and they will stay there because they're being held in their called into the tissue. And trying to understand again whether the T cells in the brain, especially the CD8 T cells in general, could be driving the disease. We ranked all expressed genes in the T cells in wild types and knockout animals. And you can see that the T cells in aging animals are activated. They have certain genes being expressed going towards T cell differentiation, lymphocyte differentiation. But what you do not see in the controls, but what you start to see in the knockouts is that they also have genes expressed that fall into the terms lymphocyte proliferation, cell killing, and response to interferon gamma. So really, again, confirming this kind of heightened immune response in these CD8 T cells. And then finally, we try to look whether microglia and T cells in this model are maybe interacting, because we see this tight interaction also via immunofluorescence. And this is now a cell chat analysis. So here you have the outgoing interaction strength. So whatever population you see here that we defined, you see how what kind of signals they're sending, and this is the incoming strength. So what kind of signal are they sensing? And you can see here, for instance, the DAMs are sending something, but they're receiving quite a lot. And in a normal situation, you have a very small population of the scalar G1 PD1 positive CD8 T cells, which I showed you before, has to have this huge type 1 interferon signature. And in a knockout, these cells really increase their interaction strength. So they're really sending signals to neighboring cells. And what they're sending, you can see here. So first of all, we looked into the DAMs. So this is now the sender. The DAMs are the sender, and either the effector CD8 T cells or this type 1 interferon CD8 T cells are the receivers. And you can see, as expected, MHC2 gene with CD8, but also other like SPP1, osteofontin, which is produced by these microglia that the T cells can respond to, so being activated. And on the other hand, you have now this type 1 interferon T cells as sender, and you can see beautifully that homeostatic, intermediate, and DAMs can be receiving, and this could maybe also be the reason why even in the homeostatic microglia we see a little bit of activation or this DAM signature. And this is just to confirm again, because we do have the site-seq data on a protein level in this model. We see that also the microglia are upregulating CCR5, which is the receptor for CCL4 produced by these T cells. And we also see TIM3, which is the ALGAL-S9 this interaction here. So you can basically confirm or infer, let's say, this interaction between microglia and T cell, and this could be really a vicious cycle that will promote and degeneration in this disease. And with that, I would like to sum up what I've shown you today. What we think is a great model to track any kind of immune cell coming into, or most immune cells coming into the brain, is the CXCR4 model. So you can really fate map cells coming in. And of course, you have T cells that can proliferate, that have different T cell receptors. And you have normal physiological aging with a little bit more T cells in the brain. However, in the SPG15 knockout, and we don't really know the mechanism in that sense, but you can imagine that if the lysosome is not really fully functional, especially in microglia, that this could have originally an effect on these cells. And what it leads to is to an increased interaction between microglia and T cells, and also shift towards a disease-associated macrophage signature to expression of MHC2, maybe giving T cells a platform to interact with, and to an exacerbated neurodegeneration due to neuroinflammation. And with that, I would like to thank especially Mark Bayer and Ralf Sturm, who initiated this study, without whom this would not be possible. Mark, who is doing everything on the T cell side, Of course, my lab, who supported us throughout the story and performed the experiments. And I would like to thank you for your attention. I'm very happy to take questions.
Thank you, Elvira. This was a great talk. So I'm gonna hand it over to Gloria to handle the questions. Just be reminded that you can ask the question yourself. Just raise your hand in the chat and you can ask it, or Gloria will read it for you. Thank you very much.
All right, thank you for the talk. We have the first question. The first is, your omics showed microglial MHC2 induction, but you see the greatest expansion in CD8 positive T cells, not CD4 positive as might be expected. Do you have any idea why this might be?
So this is a question from Dominic Simpson. Thank you.
Yes, so I think that yes, so MHC2 interacts, of course, with CD4, but I think the expansion itself is likely not driven by MHC2 expression. I think that it may be not related completely because, I mean, macrophages express MHC2 probably a little bit all the time because you are, it's an antigen-presenting cell, but you have this like huge increase. I don't think that the expansion of a T cell will be necessarily driven by MHC2. Oh, sorry. but yeah, we can celebrate.
That means cheers.
Yes.
Cheers to yourself. Congratulations for a great talk.
And yeah, why certain areas of this microglia start to express so high levels of MHC2, we don't know, but I think the expansion is completely not related. But we also see CD4 expansion as well, right? It's just the CD8s are probably the more neurotoxic. In that sense, we see also CD4 positive Treg expansion, which is likely to try to kind of shut down, the immune activation, but they may not handle it very well, and that's what we see there. But yeah, I don't think that this is related.
Right. The second question from Dila Sugunekaya Sinar, I hope I pronounced it correctly, says, thank you for the fantastic talk. Did you observe any difference in the T cell population at the periphery and or circulation in general? And then did you see any changes in microglia antigen presentation in functional microglia antigen presentation in functional level, or any changes in cytokine at T cell recruitment level, CCL3, 4, 5?
Yeah, so we did check the periphery, the thymus, also the blood. We did not see big changes there in terms of populations, not at least that we, we plotted everything and we disregarded the idea that something's changing already in the periphery. But this, maybe we have to look into that a little bit further, and not only the thymus and the blood, but maybe also other tissues in there. And in terms of production of cytokines, I mean, what we see here, for instance, you have CXCL16 being expressed, and for the SPG11 story that I've shown you, so the people made the effort to produce iPSCs and make microglia out of them, and then they also see some cytokines that could attract T cells into the brain. But in our particular model, we did not kind of isolate cells and look at what they're making in that sense. We only have this transcriptomic data and the surface marker expression in this sense. But I would assume that they have to call for T cells, because I think that this kind of entering into the brain and randomly remaining there the signal must come from someone. Now it can be, of course, also other cell types, but we don't have data on this.
Next question comes from Giulia Albertini, who asks, thank you for the very interesting talk, and asks, Holzmann's lab demonstrated that in FTD, microglia recruit T cells, and the number of T cells correlates with the extent of neuronal loss. Have you addressed a similar correlation in your model? Additionally, have you tried inhibiting interferon signaling to see if it could prevent T cell expansion or mitigate neurodegeneration?
Yeah, excellent questions. So no, we have not assessed like a line, but you can see by the behavior also that's worsening, that you could kind of, infer that it's worsening with time, because you have already at this 2 to 3 month old animals where you do not have behavioral deficits, you already have a slight increase of T cells in there. No changes in microglia at that time, right? We don't know transcriptionally, but on a protein level and population level, nothing. So I would assume that there is a linear progression in terms of T cell numbers, but we did not assess this. Also, breeding these mice is just ridiculous. I mean, making these mice and letting them age is a little bit tough. And no, we did not try to treat them with block any kind of pathway. And this would be super interesting to do, because I think this is the next step to really try to also help the patients, right? Try to understand the molecular things or cells that play a role here. For SPG11 mice, recently people also came up with T cells, but this is a little like for the SPG field, more or less new, that microglia T cell and interaction. I think this is a very good translational approach for the future, yes.
Michael Sio asked, said, very interesting talk. Did you see any alteration in BAMs in your model? Given the known role of BAMs in antigen presentation, do you also think that this could be interacting with T cells in your SPG15 knockout model?
Yeah, we looked at it briefly, but we did not have at the time, because back then, this was 2018, and I was running the experiment still myself. Not anymore, unfortunately. We did not have all of this BAM subpopulation markers in there and everything. So we did not see a huge increase or anything. But from the picture that I've shown you in the beginning, I would assume, let me just pull it up here. I would assume that in these animals, the BAMs and the meninges are, if they have this high MHC2 expression, and there probably it really makes a difference for the T cells because there are many. I would assume that also the meninges changes, but I have to say that prepping these brains, most of the cases we took away the meninges, because back then it was not that important. I mean, I think the work done in this field is quite new. We did not look into that deeper, but yeah, it could be that BAMs are as important as microglia.
All right, and next, Johannes Szlachecki, I guess, says, wonderful talk. Can you detect axonal or synaptic degeneration? And do you see microglia T-cell myelin interaction? Do microglia express SPG15, and do you suggest that this is a microglia autonomous effect, or is it signals from the environment, like neurons, that activate microglia?
Okay, I try my best to. So yes, microglia do express SPG15 a little. No, it's not the main expressing cell type in the brain, but they do express it. And I think also that's the reason why we see this late onset neurodegeneration in these animals in the brains, because I think it's an accumulation. Let's say your lysosome are not working, and then it takes a lot of time because the machinery is huge, right? I think there's a lot of redundancy there, but it will take some time. We know that SPG15 or SPGs can be also induced by interferons. So the interferon gamma will induce SPG11, for instance. So it could be that under steady state, and this is the human cell atlas and the mouse cell atlas for the brains, that we do not see high expression under steady state, but any kind of perturbation could lead to higher expression. Now, axonal degeneration, we looked only in the spinal cord. So yes, we do see already spinal, the motor neurons are gone, but in the brain itself, we are trying to look at it right now. We don't have the data yet, what's happening to the synapses and everything. I think this is something that we did not address just in new end, but I could also imagine that if microglia are, shifting towards this kind of phenotype, that also the plasticity of the neuronal network will be impaired already at that time. And then there was another question. I don't.
Yeah. I see that there is another question.
Oh, a question.
I don't know if I can unmute.
No, I think in the list of questions, I did not answer all of them, but maybe, yeah you can.
I think there's one from Oleg Butovsky. He has his hand raised.
I did.
Yes.
Elvira, beautiful talk, and there's no question how much T cells are important in microglia regulation. So we're trying to get out of brain box and look at peripheral cells. You narrowed down nicely to CD8 microglial axis, and as well you showed some interesting connection between galectin-9, and other interesting checkpoints. My question, what do you try to manipulate of this axis? And extending the previous question, not necessarily PG15 expressing microglia, what about CD8 T cells? And again, if you narrow down to this crosstalk interaction like this, we try to manipulate and see where you can at least affect microglial phenotype and functions.
Yeah, it's really, I think, a fantastic way to go about it. It's would be the next step. We did not interfere with these pathways at all so far. So we have tried to characterize it, and I think we need to do some more, let's say, functional assays maybe as well, ex vivo, to really make sure we are on the right path before treating old mice or trying to. I mean, reviewers asked us to knock out T cells, to knock out microglia, but I don't think that's the way to go. I think we need to go into these pathways.
I would not knock out if you want to. I make it a simple comparison. If you want to learn hard, you want to get out of hard, you need to learn it in vivo. So
yes, no, I agree. We so yeah, I think interferon itself and also the TIM3 interaction could be something. But I also saw a recent paper on the CCR5 microglia expressing CCR5 also playing a role in activation in a different context. I think we have different pathways, but we did not touch on them yet. Happy to see this maybe also done in other neurodegenerative diseases, because the pathways are quite similar to, you know, Alzheimer's or any kind of other disease you see.
Yeah, all right, thank you.
I have a question, but it's more like speculation. So first of all, wonderful talk, a really interesting amount of data. I really loved all these things about the local clonal expansion of T cells. I think is super cool. My question is maybe not directly related to this. It's just more like speculation. So, so I could see this, like in this image that you have only a subpopulation of microglia expressed in MHC class 2. Are those the ones that are interacting with the T cells? Or maybe is MHC class 2 doing something else in addition to antigen presentation and driving the clonal proliferation? What do you, this is pure speculation, but what do you think?
So the answer is no. MHC2 positive cells do not always have a T cell there. When you have T cells on MHC2 negative cells as well, also CD4s will interact with MHC2 negative microglia. So it's not a complete, I think the activation will happen through other interactions as well. It's not necessarily antigen presentation there.
Right.
Because I mean, what are you gonna present? Because you don't have degeneration yet, and no demyelination yet. So I think something else is happening first, and maybe it's a preparation then, or maybe these are the cells that maybe are taking up already synapses, or something is happening there. It could be, but there is no correlation so far, besides the one transcriptomics, between the MHC2 and the T cells. In general, it just led us kind of, it was like, okay, let's look at this, and then, oh, okay, let's look at the T cells, but I don't think it's like on an antigen presentation level completely related.
Thanks. Do we have any more questions from the audience or from the organizers? No? So then maybe we can say goodbye to Elvira. Thank her very much for an amazing presentation and incredible amount of data. If you want to turn on your cameras, you can say goodbye and thank you to her. Yay, thank you.
Yeah, thank you so much for all the questions, which I think is an inspiration also for us to continue, you know, working on the mechanisms in more detail.
Yeah.
It will be fantastic. Cool data. Thank you so much.
So we'll see you guys in a month. Thank you very much for attending, and just remember that we can sign you up for the emails if you want to. Thank you. Bye bye. Thank you, Elvira.
Bye.
Thank you. Bye.
Thanks. Bye, guys.